Despite the current situation with the pandemic and other crises, the ever ongoing effort to place humans on Mars is still continuing. Some billionaires of today's world are saying that the idea of colonizing Mars is close to possible and can be achievable in a few decades. But what can be done in just 30 years that will bring us to a whole new planet? I asked myself, what technologies? Is it using renewable energy to provide electricity? Or is it building those extravagant white domes on Mars while also living on life support systems since our bodies are not adapted to the environment there? Well, one of the answers might just be something that has been in living organisms for billions of years: DNA. Let's first discuss why humans should at all colonize Mars. Well, first things first, with the ongoing pollution, deforestation, climate change, and many more, seeking a second home for mankind should be focused on. Two, we are creative and curious species at heart, so there is more good than harm done with us trying to colonize Mars and our efforts to do that in the future. Now, going back to my talk on DNA. What does DNA have anything to do with bringing people onto Mars? But actually, with DNA, literally anything is possible. If done correctly, we could potentially make our bodies adapt to life on Mars. For one, we could design specific genes in the human genome to be resistant to radiation, live with a gravitational force that is specific to that on Mars, and even be less dependent on oxygen. Potentially, we would be creating a superhuman. This might be a real life superpower of some sort in the future. One of the ongoing problems we face today on getting people to Mars is the inability to withstand harsh and unusual environments there. Mars barely has an atmosphere, nor does it have any such a magnetic field. These are some of the characteristics that greatly differ from planet Earth. So, if we do manage to get someone to Mars, we'd most likely be putting them at risk. Unless major and extreme measures are taken, their cells would not be able to withstand the radiation on Mars for too long, causing their cells to constantly repair the breaking DNA. And this constant repair would result in numerous mutations that can be fatal to the person as they develop diseases like cancer. This may sound like a pretty extreme example, however, there's no saying what will and what will not happen. So, what can we do? Our body needs to adapt to a lot of things and factors on Mars. So, let me tell you in this talk about the current researches that could one day be used to address these problems and what you can do as well. This is also where gene editing and gene therapy comes into picture. Gene therapy and gene editing is an emerging technology that is being used to treat various diseases. Um, CRISPR-Cas9 falls under this gene therapy category. It is a fascinating tool that might just be the icing on the cake to get our species to the red planet. CRISPR-Cas9 is a system that enables medical researchers to edit parts of the genome. They can remove, add, or alter sections of the DNA sequence. It is currently the simplest, most versatile, and precise method of genetic manipulation in the science world. In more technical terms, scientists can create a small piece of RNA with a short guide RNA that binds to a specific target sequence of DNA in the human genome. This guide RNA also binds to a Cas9 enzyme, in which the process of CRISPR takes place. Potentially, there are virus delivered gene therapies, microbiome, epigenome therapies that can be done. The Wang lab, led by Harris Wang, aims to have kidney cells to make 9 amino acids that our bodies cannot biologically make. This would involve creating around 250 new genes and embedding it into the human genome, another way CRISPR-Cas9 comes into play. Moreover, this would mean the astronauts traveling to Mars would not need large food supplies, since a small intake of sugary water would be enough, hence making life on Mars more plausible. This would also allow fewer items to be carried on rockets to planet Mars and make life more resourceful there. Another important finding was done by Takuma Hashimoto, a team of researchers who showed that an extreme version of tardigrade's genome can improve radiotolerance in human cells. So tardigrades are small aquatic animals, which means this type of method is similar to CRISPR-Cas9, but uses a gene from another organism instead of us humans designing or editing one. Additionally, the Mason lab has developed an artificial construct of the p53 gene that is involved in preventing cancer. He hopes to insert this construct into human cells, preventing the cells from developing a form of cancer. With this, theoretically, those large spacesuits might not be necessary to wear on Mars. With all this ongoing research and many more, it is safe to say that gene therapy may make us more fit for space, allowing for travel from Earth to Mars possible. However, assuming the ethical problems of gene editing are resolved, would it be humans that are going to eventually colonize Mars, or will it be an entirely new species that emerge from humans? CRISPR-Cas9 is widely used now and is targeted towards making humans more adaptive to physical environments on the red planet. So the question now is not how we're going to colonize Mars, but who. Now, what can we all do to facilitate this mission and possibly achieve our goal in the near future? Well, we can obviously start with our own ideas. Think of ways to build energy efficient or cost efficient domes that can be built on Mars. We can also help fund projects that are working on these emerging technologies and also towards gene therapy and gene editing to achieve qualities that can help humans. This goal is still in the future, but I think with us coming together, we can achieve the goal of colonizing Mars and officially taking our species to another planet. My name is Dharssheni Senthil Kumar, and thank you for listening.